Let's go back in time a bit. Of all the methods of chemistry research you have chosen to specialize in, how did you become interested in doing research with mass spec? And what do you find compelling about the field?
Okay, well actually I backed into it because I was originally working on nuclear magnetic resonance, NMR, which tells how atoms are connected in molecules. And about that time, NMR had just been changed by introduction of Fourier transform way of doing the experiment. That later got the Nobel Prize for Richard Ernst, who was at Varian at the time. And so I learned about NMR, and it has a lot of similarities to one of the kinds of mass spectrometry, which involves a small cyclotron. And so it occurred to me that maybe somebody should apply the Fourier method for NMR to the cyclotron, and that's when I got together with Mel Commissaro, who was the co-inventor of this technique that we'll talk about.
You are most widely known as the co-inventor of the FTICR-MS. In layman's terms, describe the method and how it has contributed to the evolution of mass spectrometry.
Okay, well, the simplest way to think about it is if you think of a prism, it spreads out the visible colors to give you a spectrum. A magnet does that for the kind of signals that we observe. So the bigger the magnet, the better the spread. So we use a magnet to spread out the signals for molecules according to their mass. The heavier they are, the signal shifts more in one direction than the other. And so it allows us to identify molecules because if you weigh a molecule accurately enough, you can count the atoms in it. We can tell you how many carbon, hydrogen, nitrogen, oxygen are in that molecule. And so we can do that for as many as 100,000 different molecule chemical formulas at once. So it's a way to do that. And what the Fourier transform does is instead of interrogating by sweeping slowly through that spectrum and seeing what's at each color, we get the whole spectrum at once. So that's what it did for the field. Made the experiment about 1,000 times faster.
Tell us about the High Field Magnetic Laboratory at Florida State and what impact it has had on the kind of science that can be done.
Yeah, this is, well, we use big magnets, so that's why about 20 years ago I decided it was time to move to where the big magnets were. So this is the world's highest field magnetic field laboratory. The mission is to design, build, make available the best magnet. So best magnet can be highest, strongest field, biggest bore diameter, most uniform spatially, shortest pulse, biggest gradient, whatever. And pretty much the best in the world of each one of those is in Tallahassee, where the magnet lab is. They also build magnets for other labs in the world like CERN and Argonne synchrotron facility and places like that.
What does it mean to you to be the Michael L. Gross Lecture Awardee, especially since you have been awarded 35 named lectureships and given over 650 invited lectures?
Well, this one's special because I know Mike for about 35 years, and he was the first group to try to do our experiment, this FTICR experiment. We started in Canada. He got the first funding in the United States, or anywhere really, to do that. So we've been connected for a long time, and it's special to have this particular recognition.
Some say the Nebraska Center for Mass Spec, with its roots as one of the original NSF regional instrumentation centers, has played a pioneering role in the development of tandem mass spec and FTMS, and then in the study of biomolecules. Give us a sense of the historical significance of the center.
Well, this center got started as instruments started getting more expensive, and they were so expensive that you couldn't afford to buy one for everybody. So the NSF set up this one and I think 4 or 5 other centers around the country for that kind of instrument. Magnetic resonance was another type, and this one was the main mass spectrometry facility. And one of the first things they did was establish the world's best way to detect polychlorinated biphenyls, these PCBs that come when you, from old transformers and things like that. They're very bad compounds, and this place came up with the best way to analyze those in the smallest amount. And it became a standard. The EPA standard for measuring those is now based on the Nebraska approach. And they're evil compounds. These guys had to wipe down the walls every so often and check to be sure that they weren't poisoning themselves. So that was good. And then they also were very early into what are the modern ways to do mass spectrometry, especially applied to biomolecules. So this place really did a lot to put mass spec on the map.
What do you see as the frontiers of mass spec, And particularly, what sorts of new questions will we be able to address at the chemistry-biology interface with innovations in mass spec?
Okay, I'd say the number one area is what people call proteomics. And people have heard of the human genome. That's DNA. There are about 20,000 genes in your body. Each one of those makes one protein. But there are a lot more proteins in your body than 20,000 because they're modified after they're made. And so the next level of the genetic code will be to identify those modifications and what they do. They're what make different cells different from each other. They change in disease versus normal. They change in different kinds of people. So that'll be the big frontier. And the genome costs $3 billion, the human genome. This project will be that scale of activity. The other general area is looking at complex mixtures. For example, petroleum is the most complex mixture in nature. That can have 100,000 different chemicals in it. We can see all of those at the same time and identify them in a single mass spectrum. So areas that would apply to that would be environmental analysis, also metabolomics. If you want to know all the chemicals that your body makes, all the small molecules, you'll have to sort through thousands and thousands of those. And mass spec is going to be the best way to do that. It's the most sensitive of any of the analytical techniques for that purpose.
What advice do you have for the next generation of scientists?
Okay, you can ask an academic to pontificate is irresistible. But I would say a couple of things. One, the most important is you have to enjoy what you're doing. If it's not fun, you're not going to do well at it. And I know that because the first project I had as a graduate student was one that I didn't like. And my response was I didn't do anything for about 3 years. Then my wife got pregnant and I decided I better find something that worked. So I switched projects and things worked a lot better. Another thing I would say is don't try to do something because it looks easy. Everything important turns out to be hard anyway. So you might as well work on something important. So those would be the two main guidelines, I think.
Well, you have done amazing work and we are all inspired by you. So thank you for joining us today.
Thank you.